that's your peroxisome. Food vacuole. We are now entering the ones that are only in animals. A food vacuole is only in animals. And its job is to store food. It's like a pantry. It takes the extra food, puts it in there. The cell can call on it when it needs it. The lysosome is the next organelle that is found only in animal cells. So lysosome, you can say only in animal cells. It also functions to clean up, much like the peroxisome is going to do. Usually I have students think of Lysol, the cleaning solvent. So lysosomes, like Lysol, the cleaning solvent, its job is to actually break down food into digestible bits and, make things nice and tidy. Inside of the lysosome is a particularly acidic environment. So inside the lysosome is particularly acidic. That's a good thing because it allows for all of the bonds that are holding the cellular food together to be broken. It decomposes it. Anytime we have a cellular or any chemical reaction that results in a breakdown of something, it's called the decomposition reaction. And that's encouraged or sped up by highly acidic environments. So that's what we have in the lysosome. Next one that is plant, or that's animal only, we'll do together. The centriole and centrosome. A centrosome is made of two centrioles, and that's why we do it together. Centrosome is made of two centrioles. And I draw them this way because they are two things that always occur at right angles to each other. So the whole complex of both of them is called the centrosome. One individual piece is the centriole. Now, this is a slightly inaccurate picture because if you were to encounter a diagram, this would look like, my usual comparison for students is a Twizzlers pull and peel. It's like a small packet of microtubules all lined up, like if I had a bundle of straws. Other times, textbooks call it star-shaped. Sometimes it's referred to as like churro-shaped. So it has ridges on the side, churros like covered in cinnamon sugar, you know what I'm talking about? Some people are like, what? And I'm like, when was the last time you went to a parish festival? Come on.
Will you ever have like 3 centrioles together? Or could you have like 2 sets of centrioles?
The reason I say no is because of their function. The job of the centriole individually, they migrate to opposite sides of the cell during cell division. And there are only 2 poles of the cell. So any cell will only ever have 2 centrioles. Their job is to organize cell division and make sure everything gets divided where it belongs when the cell is completing mitosis. You'll become very familiar with their functions later. But for those of you who know the mitosis song, you're probably now singing it in your heads. For those of you who don't, it's okay, you'll sing it later. I can't wait. I love sing-along hour. That concludes all of your animal cell-specific organelles. Next up are plant cell-specific, and we'll do them nice and quickly because one of them you should already know about. But I'm not going to skip ahead because I think it's out of order on your list. Usually when people look inside of plant cells, yep.
Do like the organelles in both animals and plants, like, do they do the same thing?
Yep. So their functions will all be the same if they're in both. The central vacuole, if you look into a plant cell or look at a picture of a plant cell, it looks like there's this big bubble or balloon that's empty. It's not actually empty. It's the central vacuole, and it contains a great deal of water and dissolved minerals and nutrients, ions, things like potassium or sodium. The cell keeps them there for storage purposes. So consider this like a big liquid storage, giant liquid storage. And it serves, besides the function of storage, it makes plants rigid. So it makes their cells firm. When a plant cell and its central vacuole empties of water, you've seen it before, the plant wilts. When you water it and the plant reflux, it's because the central vacuole has refilled with water. So that particular action is called turgor, Turgor, and it's what gives plants structure.
How does something filled with water give plants structure?
Think about it like a waterbed. If the waterbed didn't have any water in it, it would just be like flat, right? But if you fill it, it's going to have the shape of the bed.
So it's like the membrane. Oh, it's like the membrane around it called the center, and then.
Well, the actual thing is called the central vacuole. The membrane around it is actually called the protoplast, of all of your names.
Oh, what's turgor?
It's the pressure that results from water filling a plant cell. Turns out, though, you know how I said you can refluff the plant that's wilted? If you've emptied its central vacuole by forgetting to water it, you can refluff it. That only works if you don't let the whole cell dry out. If you let it go too long, it's going to die. So, you know, if that's your chore at home, like watering plants, you might want to. A few days is okay, wilty, but more than that, probably gonna kill it. That's no good. What's chloroplast is next on your list, right? I'd rather do cell wall after this, and then chloroplast, but we'll go in order. What's chloroplast do? The site of photosynthesis contains a compound called chlorophyll. It's what makes the plant green. And it actually has a couple of funny, peculiar parts. So in the chloroplast, inside of a chloroplast, there's some individual little parts, so it's like an organelle, has organs of its own. They're called the thylakoid and the grana. They're small stacks, like discs. They look like stacks of Smarties all lined up next to each other. And that's actually where part of photosynthesis occurs. It makes it go faster. So thylakoid and grana. Plasmodesmata. Is that next on your list? Excellent. Plasmodesmata are just holes. Yep.
What are the two things do for chloroplastia?
They're the parts inside the chloroplast. So it's like, it's weird because we think of organelles as being the smaller part inside of the cell.
But those were the ones that was like on its own and then it was part
Yep,
of
so
it.
it has other smaller parts inside of it.
Well, what do they do for it?
It actually allows the photosynthesis, a site for it to happen. So remember how we said that clay allowed particles to concentrate and its concentration made things go faster?
Okay.
These allow the necessary chemicals to concentrate and thus aid the process of photosynthesis going faster. But those two parts are why we believe the endosymbiont hypothesis. Because for a subpart to have subparts, then that subpart with subparts must have at one point been an independent thing. Plasmodesmata, really much simpler than the name sounds. It's tiny holes in between cell walls that allow communication from one cell to another. So like tunnels, there's little tiny holes. Allows the passage of materials, minerals, anything that needs to go through, goes through those holes. I know, you're like, it sounds so fancy. We do that sometimes. Cell wall, plant only, super, super important. really, really important. And we're gonna use our cinder block walls as an example, because I don't know about you guys, but I don't have a degree in engineering. Structural engineering, not my thing, but I can look at that wall and understand this. So the bottom set of bricks of my cinder block wall behind us bears the load of every brick on top of it, right? And so those ones on the bottom had better be structurally sound. Now, not to freak any of you guys out who sit in the back, but as physics progresses through the rest of the year, you're going to hear an awful lot of bowling balls hit those bottom cinder blocks.
Yeah.
So, if cinder block, if those bowling balls hit that cinder block, and in any way, shape, or form damage this cinder block, what's going to happen to the wall? It tumbles, right? It's the foundation of that wall, as opposed to if you look up at the top where that air vent goes through the room, and why we can actually hear Mr. Nanini's class so very clearly sometimes, there's cracks up there, right? There's like holes up in those that, anybody like, hey, I could remove that brick. Yes, we could. We could pass messages in bottles by pulling that brick out. Why, I don't know, because my brothers walk next door. But it would be entertaining for a few moments. Are we worried about those being cracked? I'm not worried about it. I mean, okay, maybe if I were you and sitting underneath the one that looks like it's going to fall out at any given point in time, I might be worried about it. But at the top, it's not load bearing, so its structure is really important. Plant cells have to have something that is rigid in structure because the cells at the bottom have to bear the load of every cell above it. In plants, the thing that does that assists in bearing the load, is going to be the cell wall. If they didn't have a cell wall, the ones in the bottom would simply explode. I mean, that seems like it might be bad for the purposes of being a plant. Now you're like, wait a minute, we're multicellular. How do we deal with this? Oh, what's the cell wall made out of in plants, by the way? Cellulose, or indigestible fibers produced by plants. It's structural polysaccharide. Animals don't have the benefit of a cell wall. Instead, what we have inside of our cells is a structure called the cytoskeleton. Now, the cytoskeleton is not like the kind of skeleton we think of. When we think of skeleton, we think of very hard and rigid. You should think of your bones. I can't take this bone and bend it, can I? That's where a cytoskeleton is slightly different. The cytoskeleton is more like cartilage. If you were to find a part of your body that's cartilage, so the two easiest ones for you to currently find will be your nose or your ear. If you were to find it and try and bend it or squish it, squishy, right? However, if you let go of it, what happens? Right? None of us can change the shape of our nose by squeezing it or holding it for a little while, right? It's going to go right back to shape. That's like the cytoskeleton in an animal cell. It's flexible, it moves, it accepts pressure because it's made out of two flexible parts. So, skeleton. It's not technically considered an organelle, but it seems to me to be like often kind of sort of important. And it often gets skipped. Like nobody tells you about it. And that seems silly, especially for students who shouldn't connect dots. Like how can we be multicellular if plants can be because they have a cell wall, but we don't have a cell wall? How does that work for us? I assume you guys make those connections.
Wait, is the cytoskeleton only in animals and not in plants? Or is it?
A plant will have a much simpler cytoskeleton.
Okay, but they both possess them.
So for cells that move, there are two things that are going to help with movement. Either you have a flagella, actually it's a flagellum. That's our whip-like structure that spins like a propeller and will propel a cell forward. Sometimes there are certain protists who have two flagella. If they are on opposite sides of the cell, then it can direct the movement very specifically, because if you turn on one propeller, it's going to move one way. If you turn on the other propeller, it moves the other way. And if you turn on both propellers, it will move in a median direction in the middle. Other cells, rather than having a flagellum that rotates like a propeller or a whip, they're going to have cilia. They're fine hair-like projections on the outside of the cell that wave back and forth in unison in response to the cell's environment to be able to move it in a given direction. Those should be all of your organelles. Have it on fairly good authority.
Are the flagellum and the cilia organelles?
They're technically considered organelles, yes, which is odd because you're like, I thought organelles occurred inside.
Is like only one of them common to plants and one of them's animals?
Plants will typically not have either.
So they just don't move. Oh, well,
I feel as though you may have answered your own question. They rely on their environment to move.
why would a cell have flagellum or cilia specifically?
Because of the nature of how it moves, certain protists will have one over another because of having fine movements versus gross movements. So if it's something that lives in an environment where a small shift in its environment or space makes all the difference in the world, they typically have cilia. Flagella are for things that move a lot. I should tell you, flagella and cilia are usually, actually are always, limited to the cells that are in water or are aquatic in nature, because that's how they move, is by pushing the water in one direction or another. Keep in mind, water means like frog blood cells are ciliated, have cilia on them. Why? Because blood is a liquid, right? It's water-based. So that's how it's going to be able to move through the frog.
So you said most plants don't have them. What about ones that move, like the Venus flytrap or something?
They actually move from a weird series of ion exchanges. It's really quite as simple as that central vacuole filling and unfilling. You guys ever seen The Sensitive Plant, Ticklish Tim? Really? Okay, some of you have. That's good. There's a plant that if you touch it, it folds up, it moves, like it responds to touch. And that's how it responds to touch. Your touching it breaks the turgor pressure, causes all the water to rush out of the cell, and then it folds up. When it refills, it opens back up. It's quite entertaining. I always had one on my front porch when I was a little kid. My mom planted one every spring. And it's a wonder how I ended up being a biologist. Go figure. Thanks, Ma. Do you know what this thing is? What is it called? Oh, delightful. That's why I'm not spending any much more time than this on it. What's this part called? Eyepiece. How do you carry it? One hand on the base, one hand on the arm. What are these things called? This is where everybody makes up names. They're like, I don't know, the spinny thingy. The objective lenses. So we have an eyepiece, objective lenses. The short one is low, the longer one is medium, the longest one is high. I have 4 on mine because it's a a higher resolving microscope than yours will be, so I have low, medium, high, and extra high. Or low, medium, extra medium, and high, whichever one you prefer. How do you put it down on the table? All right. What direction does the arm go? Arm to your tummy. So when I place this down on the table, if I'm doing it arm to tummy, it's facing this way. Arm to tummy. Now you're like, how do you look through it? That's how I would look through it. I had it bent because I can attach it to my document camera. Please do me a favor tomorrow. You guys are at the end of the day, so it doesn't really matter for you. This is a bigger deal for first hour. When I give you one of these tomorrow, I don't want you to leave here with a headache. So this is not how we look through a microscope. What that does is forces your orbicularis oculi closed, and it ends up giving you a fabulous tension headache. It makes this hurt, which causes tension through here, which causes tension up through where those ligaments attach at the top of your brain, the top of your skull right up here. Tylenol can't help with that. Tension headaches are miserable. So what you're going to do instead of scrunching one eye up is look with both eyes open. Now some of you are like, I can't make my brain do that. My brain will be looking and what do I do? Deprive your eye of the sensory experience it requires for vision. It's magic. Try that right now. See what happens. You're like, your brain started looking with only this eye. You didn't even tell it to do that. Turns out your brain is pretty smart all by itself. It doesn't require you to think. Think about that for a minute. So do me a favor tomorrow when you're looking through it. I am going to remind you that you're all pirates. Put your eye patch on. And that eye patch is going to assist you in being able to look through it without getting a headache. Any rules I should know for how to focus? Because I was told, first hour was like, we used those before. All right, show offs. Tell me what you know.
So you would adjust it out and then switch them and then adjust it back in? Because I know someone broke a microscope. Does it like went into the screen?
So what you're going to do is when you put the slide on it, stage goes all the way down. Stage goes down. And then you're going to move the stage all the way up and focus so the stage moves down again. So the stage is down when I put the slide on it. I'm watching to make sure my objective doesn't hit the stage, and I go all the way up. And then I focus, looking through it, like that, away so the stage moves down. The objectives are the most expensive part of the microscope, so we never want physical contact between the stage, the slide, and the objective. These are what costs us serious, serious money. That also brings us to the point, which knob do I use to focus on low power versus the knobs I use to focus on high power?
Right.
Turns out both knobs are on both sides of the microscope. Mine combines them. My coarse adjustment is the big one, and my fine adjustment is the small knob in between. Your microscopes are going to have two different knobs. A big one, coarse movements, big movements. That you only use on low power. Let me repeat that to make sure all of you who are not paying attention to me heard it. Only use coarse adjustment on low power. You don't want to use it when you have a higher objective lens on because it moves the stage. Anytime the stage moves, you have a possibility of the stage and the objectives making contact. When switching between low and high power, do not unfocus your microscope. Has to stay focused and in the center of your field of view. Because if it's not focused on low, it will never focus on medium or high. No matter how hard you try, you spend the rest of the day sitting there fiddling with the fine adjustment knob. It's not gonna work.
Which knob is which again?
Big fat one is coarse, that's the one you use on low. Little one is called fine, and that's the one you use on medium and high. Turns out the microscope, besides those very basic rules, when we store it the stage all the way down and on low power. No slides on it, and then we cover it. Besides those rules, most of the microscope is all about experience. There's not much point in me telling you how to use it because it doesn't mean anything until you touch the microscope. So I'll tell you now, tomorrow when you're in lab, you're going to struggle a bit to find things. You're going to struggle a bit to focus. I will gladly come help you, but I want you to struggle first. Not because I take delight in your struggling, but because you're not going to learn anything from watching me focus a microscope. Turns out, I've spent years focusing microscopes. I'm capable of doing so. Only way you're going to be capable of doing so is if you work on it yourself. So keep that in mind. I'll gladly help again, but not something that you're going to be appreciative of. Tomorrow, I also must tell you, please do not plug the microscope directly into these. You'll get a surge protector, so then the table of you can plug yours in together into one. You guys don't call them surge protectors, you probably call them power strips. You guys will plug all of yours into one that way. We're going to have you guys join a larger table or work at the end of the table. So you're not like hanging out working on your own table. Yep. I'm going to ask you guys, please, tomorrow when you're working, be careful. These microscopes are between $300 to $500, depending on which one you get. I would prefer you not break them, because that makes me cranky. Turns out, a little bit. So, don't do that. First hour said, if we make it to our candy collection, does Open Note include open worksheet? Sure. Why not?
This is our homework.
Yes, this is your homework.